Congratulations to all these very young and very talented young investigators. Now it's time for discussion and questions. Please.
Hi, question for Gemma. I'm with my PSC hat on. I mean, we often see a lot of senescence in the biliary epithelial compartment. Have you thought about senescing your cholangiocytes before you co-culturing them with your malignant cells to see if there's, differences or how that plays out? Does it amplify the situation?
That's actually a really interesting question. No, we haven't thought about senescence in terms of a co-culture model. But
Yeah,
maybe that's
just
something
might
we
be an
should
interesting
look at.
experiment to do, so if it amplifies what you find.
Let me answer your questions. So, Gemma, you need to mention about the culture conditions. It's mimic the senescence where you grow the cells in the 0.1% BSA rather than full median. So it's mimic senescent status. Then my question to, I can't remember your name, sorry. So KLF4, CRISPR's knockout, and affect merely 5% cell growth ability. Whereas in vivo, xenotransplantation, you can see dramatic reduction of the tumor volumes. Why they are so much different?
Sorry, could you repeat? I didn't understand the first part of the question.
So CRISPR's knockout of the KL4
KLF5, yeah?
cell lines. You look at the effect on the cell's proliferation ability, and you can see only merely 5% reduction.
Okay.
Am I right? So whereas in xenotransplantations, you get such dramatic effect. Why they are so much different?
Okay, now I understand. Sorry. Thanks a lot for the question. It's a really interesting point. Basically, I think we in the CRISPR knockout cells, we are the effects are not as huge as the ones that we observe in vivo. It is widely known that KLF5 is in some types of cancer are associated with the malignization of the tumor. So I truly think that although we see differences in proliferation and apoptosis and migration and so on, I think that by knocking out KLF5 in these cells, we are taking the capability of these cells to malignizate and to clearly being implanted in the liver. So they are losing the capacity to implant and to create novel niches to grow. I think it's not related only with proliferation, but also with the stemness capacity of these cells.
Sorry,
you go for it.
Yeah. So in your RNA sequencing data, did you see any compensatory responsive transcription factor activated when KLF4 was knocked out?
As far as we know, we haven't observed any compensatory mechanism there.
Yes, I want to welcome Alex into the Q&A. So if anyone wants to make questions also, please forward. Look.
So I have a question about KLF5 as well. So do you have any, so there are two halves, I guess. So if you silence KLF5 and then allow it to reactivate, do your cancer cells kind of become malignant again? So, with like an shRNA. So is this a very dynamic process?
Well, that's a very interesting question. We don't know that. We know that when we overexpressed KLF5 in normal human cholangiocytes, we were able to increase the proliferation and the migration and evasion capacity of these cells. But probably I would guess if I reintroduce again KLF5 in these CRISPR cells, we would be able to rescue some of the phenotype that we observed.
And I suppose that kind of links to my next thing. So have you looked at what KLF5 directly binds on the DNA? So it's a transcription factor, right?
It's a transcription factor, yeah.
Do you know what its direct targets are? Because I guess your proteomics and your transcriptomics will tell you direct and indirect.
Mm-hmm.
So do you know what it's sitting on the promoter of?
So in cholangiocarcinoma, we don't know, but in other cancers, it's reported that it regulates several cyclins. For example, cyclin D1 is known to be one of the direct target of KLF5, for example. It also regulates some of the cell cycle arrest proteins, P27, PIM1 proteins, and it also regulates some of the apoptosis related ones. So that's why we observe when we knocked out completely KLF5 in these cells, we observed at the basal condition a slightly increase in apoptosis. And we observed, for example, when we conducted the proteomic analysis, we compared the both clones with the CRISPR control and some of the proteins related with chemoresistance, we observed that the ones related with the induction of apoptosis, BAX, BAC, and so on, they were markedly upregulated in the knockout cells compared with the control ones.
It would be interesting to look at that and PRH.
Yeah. No, no.
Can you come to the microphone? Sorry, just for the people online. Thanks, Sheila.
I was going to ask you if you'd looked to see what had happened to PRH in your studies, but actually we have noted that when PRH is upregulated, we do see changes in KLF expression.
In 5?
I think in KLF5. I'll have to
Whoa.
go and check now. And we also see alterations in migration, invasion, in anoikis, so
would
in
be really-
stemness, etc. So almost all the same phenotypes, and so there's very likely a regulatory
Probably too
loop
very
or a
interesting to check.
component that we should probably go
Yeah.
and talk about.
Yeah, thanks a lot.
Well, are there available different drugs, fortunately for KLF5, not only the one that were used here? Is there any clinical trial ongoing with this? How is the potential transfer of the results? Because the effects in vitro and in vivo are very significant.
Yeah, so far when we started this work, there was only one paper published with this inhibitor in colorectal cancer. It was the only study published when we started. Now there's another one also in colorectal cancer, but no clinical trial is so far planned with KLF5 inhibitors. As far as we did the in vivo experiments, I think we should be cautious with the doses, because when we tried to replicate the doses used in the colorectal cancer paper, we saw kind of a toxicity in mice. We have to decrease a lot the doses used in mice, because I mean, if we can kill the tumor, but we don't want to kill the host. So we reduced dramatically the doses used in the colorectal cancer paper. We still observed very nice data, but I think we have now to do several tests to evaluate efficacy. And last year, a new inhibitor came out. Supposedly it's more potent than this one. We could test if the effects are mimicked and if it's less toxic, but I think it will be.
But it's very good that even if you use doses significantly lower, you have significant effects.
We are using
colorectal.
five times lower doses, and they use both twice a day, and we are only using once a day. So it's basically 10 times lower than the doses used for colorectal cancer.
I'm sorry, I've got another KLF5 question. When you present data like that, you're going to get lots of questions because people are excited. I'm just thinking now about moving this into the clinic. So in terms of patients, is it expressed in all cholangiocarcinomas, or would we have to do some immunohistochemistry or some other test to test expression in order to select for patients or enrich for patients most likely to benefit? How do you see things going?
In the patients' data, we observed that all patients displayed a very remarkable expression of KLF5, both at the mRNA level and also at the protein level. There were no differences regarding the genetic drivers, and also I didn't present it here, but there were no differences as well regarding subtypes. For example, the KLF5 was upregulated in intrahepatic, peri-hilar, or distal CCAs. So I guess that we can try to use the inhibitor for all cholangiocarcinomas, because it seems to be a general event during cholangiocarcinogenesis. Although we know, that this gene is not hypomethylated, it neither amplified. So it's not, the upregulation of KLF5 is not a result of genetic mutations directly, but might be regulated by other targets during cholangiocarcinogenesis.
We were very impressed that this a highly common event in the tumors, but in the cell lines, as you saw, the expression is huge in all the tumor cell lines that we evaluate in vitro. 8 different cell lines, all of them huge level, and the normal ones not very, very low levels. And the effect of the compound has no effect in the normals, but in the tumors is very significant. Although it's a common event in the different tumors, maybe you, Pedro, want to comment that there are some mutations directly associated with the expression, very clearly.
Yeah, basically, no mutations, at least so far for KLF5, are known, but one of the endogenous regulators of KLF5 is FBXW7, which is a protein that ubiquitinates proteins for degradation. It is a well-known tumor suppressant in a lot of cancers. There's a paper that shows that FBXW7 is decreasing cholangiocarcinoma and mutated in up to 10% of tumors. So, and the paper that came out 2 years ago from Diego Calvisi's group, where they did a Sleeping Beauty with inactivated mutations of FBXW7, they were able to generate cholangiocarcinoma tumors in mice only by inactivating this gene. So we guess that since there's a direct regulation of this protein of KLF5, one of the reasons why we think that KLF5 is being upregulated is because FBXW7 goes down, which decreases the ubiquitination of KLF5, decreases its degradation, and allows KLF5 to go to the nucleus and promote the transcription of genes. Another level of regulation could be the Wnt signaling pathway. Since when we treated normal human cholangiocytes with Wnt ligands, we were able to observe that KLF5 goes up. So probably when the Wnt signaling pathway is activated in cholangiocarcinoma, KLF5 is activated. And when we treated cholangiocarcinoma cells with the KLF5 inhibitor, after increasing proliferation with Wnt ligands, we were able to totally rescue the effect induced by the Wnt ligands. So probably another level of regulation would be through the Wnt signaling pathway.
So definitely the fact that there are different inhibitors and the relevant role of KLF5 deserves some further discussion and attention, hopefully a movement to clinical trials. I have a couple of questions also for Alex and also from Gemma. Maybe Gemma, did you evaluate the crosstalk between the normal cholangiocytes and the tumor one? Is there any evidence of the role of extracellular vesicles in this crosstalk, or did you evaluate that, or are you planning to do something?
Extracellular vesicles is definitely something that's in our mind. It's not something that we've managed to get around to looking at yet, but we're trying to prioritize the metabolism and then next the proteomics to try and get our full multi-omics approach done first, and then.
And I have a methodological question. The normal human cholangiocytes that you comment, are they primary culture or are they immortalized?
The cholangiocytes I use are MMNK1, and they're an immortalized cell.
With SV40?
SV40 immortalized.
Okay, so they are not completely normal, actually. Well, that is
normal
good.
in quotations.
Yeah, okay, very good. Gemma, very good to welcome you. I mean, no, Gemma, no. It was Alex, sorry. Very good to welcome you to the Q&A. Regarding your experimental models, the hepatitis C cholangiocarcinoma cells, so how is the direct antiviral treatment affecting the behavior of the cell individually and also its relationship with the crosstalk? Did you evaluate this further?
Yeah, I made sure I had a control for this because we weren't sure what they would do, especially to the stem cells when I did my stem cell model. But they don't seem to affect the differentiation on its own, which is good. With the HCV, I think in the H2H7 model, you get a better idea of more chronic HCV infection. So once we've treated with the DAAs, that the infection goes away, and then I can assess that. But in the stem cells, we're getting probably more of an acute infection. So we still need to tinker with that to try and get a better model out of my stem cells. But yeah, we've got an overlap of about 167 genes between a CCA data set and my data set for treated with DAAs. So it is interesting to see how it overlaps between the two cancers.
Yeah, that's very, very interesting. Congratulations for the nice job. Any other question or comment? No? So, well, I will ask for a big applause to all these very, very talented fellows. Well, thank you very much. So we move on to the next session.
Thank you very much. We'll let you go. And we go for coffee.
Some break.
We have, please, until 3:20, at which point we'll have our final session looking to the future. So enjoy the networking session.
Thank you.